our lab, one of the major areas that we work on is metabolomics. And one of the tools we use for metabolomics is NMR. So in this presentation, I'm going to talk about one of the programs that our lab developed for analyzing NMR spectra. We call it Magmet. And so this was originally developed by a previous member of our lab, Minoj. And as the current NMR person in the lab, I've, as one of my projects, picked up continuing development of Magmet. So what is metabolomics? So metabolomics is the study of small molecules that are produced or found in organisms. And so you can analyze different parts of the organism, so tissue extracts, serum, urine, and so on. And this set of metabolites, presence or absence of metabolites, or how much of each metabolite can be influenced by genomic and environmental factors. So, for example, in the diagram at the bottom, we start with DNA. DNA makes RNA, which makes proteins, and it's the proteins that perform the chemical reactions that make all the small molecules in the body. So the metabolites are influenced by the DNA and all these steps that lead to the small molecules. And as well, this entire pathway is also affected by environment. So things like diet and disease. And so metabolomics has applications in studying things like genetics, toxicology, and diagnostics for health. So there are two main methods for metabolomics. One is mass spectrometry, and the other is NMR. So mass spectrometry, often this is combined with various chromatographic techniques, HPLC, gas chromatography. So it has some advantages, such as using small sample volumes, and has high sensitivity. And often molecules need to be derivatized to be properly detected by mass spec, or separated by mass spec by the HPLC or GC. NMR is the other technique that we use. So often these are performed on whole samples without any sort of separation, or derivatization. So NMR is usually considered non-destructive. And on the other hand, compared to mass spectrometry, we need larger sample volumes, and it's less sensitive. However, NMR is considered to be more reproducible than mass spec, and it can also, both methods are somewhat complementary. NMR can look at some of the metabolites that maybe mass spec can't. So our lab focuses mainly on targeted metabolomics. So in targeted metabolomics, we have a predetermined list of metabolites that we specifically want to identify and quantify. And to do this, we take mass spectra or NMR spectra of reference standards and can compare it with our unknown samples. And so this allows us to quantitate metabolites, however, compared to untargeted metabolomics, where we kind of look at metabolites as a whole and try to pick out interesting metabolites. It's lower coverage because we target specific metabolites. So this is a picture of our NMR machine. So the NMR spectrometer is a superconducting magnet. So it's a cylinder in the upper right picture. It's cooled to 4 degrees Kelvin, and it's a 700 megahertz spectrometer. So this is kind of in the medium to high range of current NMR spectrometers. Some of them go up to 1200 megahertz now. But these are, just to give you an idea of how strong these magnets are, they're 1,000 times, 10,000 times stronger than regular fridge magnets. And MRI magnets, which operate on similar principles, these spectrometers are 10 to 100 times stronger. We have a cryo probe, which we use to excite in detector samples using radio waves. So cryo probe is different than a room temperature probe in that the electronics are cooled to 20 degrees Kelvin using helium gas, and this helps to reduce the noise in our spectra and improve sensitivity and lets us acquire spectra in much shorter periods of time. We also have a sample jet, so that's the machine on top of the spectrometer. And so this is a sample changer, and so we put our samples into NMR tubes, and then put those NMR tubes into racks, which go into the sample jet. And then the sample jet has a robotic arm that can pick up and put the samples in and out of the spectrometer. And so we can run potentially up to 100 samples a day in this way. So before we acquire NMR spectra, we have to prepare the samples. So these are the steps we use in our lab. So first step in some samples, for example, fecal samples, is we need to extract the metabolites. So we take fecal material, add water, and then mix it to extract out all the water-soluble metabolites. And then for some samples, we also filter them. And this, we use a 3,000 Dalton cutoff. And this, these filters will remove protein and lipid signals. And these signals can interfere with the quantitation that we do. So we want to remove those. And we also use a strong buffer. So we buffer samples to minimize the sample-to-sample variations in pH. So we use a phosphate buffer, so 50 millimolar phosphate for serum. Serum is already regulated fairly tightly in terms of pH, so we don't need too much buffer. And for other types of samples, fecal, urine, we use 150 millimolar phosphate. The buffer also contains other internal standards that we need for NMR. So it includes 5% D2O, compound called DSSD6, and another compound called CPCA. So once we acquire the spectra on our spectrometer, we need to process the data. And so the top figure shows the raw data. So the NMR signal is basically an oscillating signal, and it decays over time. And it contains different frequencies. And so we can extract these frequencies using a Fourier transform. And so this gives the middle figure. And then we expect all the peaks to be positively to be positive, and we want the baseline at 0, so we phase and baseline correct our spectra to get the bottom figure. We also apply line broadening, which smooths out the spectra and removes some of the noise. And we also use the peak that comes from the DSS-D6 compound and set this to 0 ppm. This way we can line up all our spectra, and this lets us compare the spectra and identify the compounds based on their position. So this is a 1D proton or hydrogen NMR spectrum of serum. So all the peaks you see in the spectrum represent some hydrogen atom in the serum sample. So in the middle, there's a large water peak, and on either end are the DSS-D6 and the CPCA internal standards. And then all the other peaks come from small molecules from the serum. So going from left to right, we have aromatic and amide peaks on the left-hand side, and then CH, CH2, and CH3 from different molecules as you go to the right. And so what we want to do is identify what all these peaks are and quantify how much of each compound is in this sample. So once we process it, we can analyze the spectrum. This is called profiling. And so what we want to do is compare our unknown sample with spectra of pure compounds. And one of the ways we can do this is manually with software called Genomics. Genomics was developed in Edmonton and is used for manual targeted profiling for NMR. So you pick an interesting peak and you can search for compounds in the database that have a peak in that region. And then you can look through the list and see if you can find something that matches the patterns that you see in the spectrum. So in this case, we find this pattern with 4 peaks in it. And then we can drag the peak until it matches with the spectrum. And so in this case, this compound is lactate or lactic acid, and it has a concentration of about 1 millimolar. So then we go through other peaks in the spectrum and try to identify those until we've identified as many as we can, and then we will have a list of all the of compounds and concentrations of those compounds, and then we can do further analyses with that data. So one of the difficulties with manual profiling is it can take up to 30 or 60 minutes per sample. So you can imagine if you have 100 samples, it can take 2 or 3 weeks just to analyze them all. So what we wanted to do was to try and automate the process. And so we developed a program called Magmet and developed it as a web server. And so Magmet can profile serum spectra in around 10 to 20 minutes. And it can profile multiple samples simultaneously. So effectively, it can take 5 or 2 minutes per sample if you have multiple samples being run. And on the right, you can see a screenshot of the main the website. So there are instructions for how to collect NMR spectra and the specific file formats you need to upload a spectrum. And once you log onto the website, you can upload a spectrum, select a few options, like what type of biofluid it is. Currently, we have serum and fecal waters. And once you've uploaded the spectrum, you can click submit, and Magmet will go through the process of processing and profiling each spectrum. So we're also, other than serum and fecal water, we're also working on adding other biofluids such as cerebrospinal fluid, urine, wine, and other beverages. So this just shows a overview of the steps that Magmet goes through, essentially the same as what I showed you in the previous slides, where we do everything manually. So Magmet automatically processes the NMR spectrum, so transforming, line broadening, phase correction, baseline correction, referencing. And then it tries to identify the compounds by comparing the spacings in between the peaks in the unknown sample and the library of spectra in Magmet. So if it finds a match, it will try to fit that compound. And so one of the things, advantages of Magmet is also that Magmet can handle some variation in peak position and still in the experimental spectra and still be able to identify and fit the peaks. And so this shows the website again. So after Magmet processes and analyzes spectrum, it will display the results on this web page. So there's a spectrum viewer on top and table of metabolites underneath. And so the spectrum viewer is interactive. You can zoom in, zoom out, drag the spectrum left and right. You can also highlight various peaks and look at their concentrations that way. And you can also move the peaks around if you feel that maybe Magmet didn't quite fit the peak correctly, and once you're satisfied that the spectrum looks good, the data can be downloaded using the download button on the right-hand side. So we find that Magmet seems to work fairly well with serum and fecal so far. However, we do need fairly good quality spectra, so low noise, and the shape of the peaks has to be symmetrical, and so the shimming has to be good. And sometimes we need to look through the spectrum and manual adjustments are needed. So behind the scenes, there's also a lot of work in implementing each biofluid. So we need to attain standard spectra for every compound that we want to add to Magmet. We need biofluid spectra to analyze and train Magmet on. And we need to identify what compounds we think are in the biofluid. And then we can add these to Magmet. And then finally, we need to optimize the fitting where Magmet searches for each peak and and optimize how it gets the concentrations. And normally we need to do this for every different spectrometer field strength. So you can't use the 700 data for, say, a 500 spectrum, and vice versa. For this last point, one of the things we've been trying is to use what are so-called spin matrices, because in theory we can calculate accurate spectra for any magnetic field strength, so 500, 600, 700, using spin matrices, which are basically tables of the chemical shifts and scalar or J couplings, which describe the position of the peaks in the spectrum and the splittings and the pattern of splittings that you see. So to do this, we need to obtain accurate chemical shifts and couplings for all these compounds. So one source of this could be from the BMRB, where there's this supplementary kind of database called Gizmo, which contains spin matrices for many compounds found in the BMRB database, which contains NMR data for small molecules and proteins. And we've also attempted to do our own fitting of our own data from our 700 megahertz spectrometer, as well as older data that we acquired for the HMD website at 5 and 600 megahertz. So this shows part of the spectrum of glucose. So in black is the experimental spectrum, and after optimizing the spin matrix data and calculating a simulated spectrum, we can get something that's very close to what the experimental spectrum looks like. So once we gather up enough spin matrices and information about concentrations in a particular biofluid, we can use this data to try to simulate some NMR spectra that we can use for training Magmet. So it shows part of a simulated serum spectrum at 3 different frequencies, 300, 600, and 1200 megahertz. And you see what the effect of going to higher field strengths is. So what happens is the peaks actually get farther and farther apart, but if you kind of plot them on the same scale, so the positions of the peak stay constant, it looks like the peaks kind of get narrower and narrower. But in both cases, the peaks get more and more resolved as you go to higher fields. So there are advantages to using higher field strengths to study these sorts of samples. So the next step would be to use the simulated spectra and libraries generated from of the libraries of compounds generated from these small molecules and try to optimize Magmet at these different frequencies using all the basically just simulated data. So in conclusion, then Magmet was developed to automatically identify and quantify metabolites in 1D proton NMR spectra. So we implemented a web interface, and we're working on implementing various biofluids in addition to serum and fecal that are currently on the website. And we've also been playing with doing spin matrix calculations and generating simulated data that might help us optimize Magmet for other field strengths other than our 700 that we have and our older 5 and 600 data. So for example, 800 megahertz, which is another common field strength for NMR. And yeah, I guess that's the end of my presentation then.
Thank you, Brian, for the nice presentation. This is very good.